we now have to do is to decode the genome, and we've kind of got it all. We've got the answer to how life works. It hasn't turned out that way. We've decoded the genome. We've decoded now, tens or maybe hundreds of thousands of genomes, and we still don't fully understand how these complex systems of cells and of organisms work. The body as a whole is nothing like a machine. Our neural circuitry integrates information and comes to a decision about what we should do. But actually that same principle applies all the way down the biological hierarchy to the simplest living organisms, to single cells.
Yes.
They too have to be able to make contextual decisions. So it can't all come from the bottom up. There has to be some way to ensure that biological organisms are genuine agents. They're not just automata programmed by their genes.
Philip Ball is one of the most influential science writers of our time, a former editor at Nature for over 20 years and written over 30 science books, many of which are bestsellers and award-winning that stretch across quantum physics, consciousness, synthetic biology, and the bizarre frontiers of life itself. Philip is back for a round 2 on the Giant Shoulder to unpack the latest biological research, which is guaranteed to make you question everything you thought you knew about how life works. Subscribe if you want to discover why life was thinking long before brains existed and why that fact truly changes everything.
Living systems seem to build in at every level a kind of fuzziness, a kind of leeway, I suppose. We can track individual cells during development and we can sort of see them almost exploring the landscape. They're going as though towards a neural cell fate, for example, and then because of the signals they're getting from cells around them, they kind of realize, to put it that way, that's not the right thing to be at that time in that place. And so they change their mind and they sort of go somewhere else. And you need that noise to have that sort of adaptability. Whereas if it was just following a program, an inevitable program to become a nerve cell, then things are going to get screwed up and things are going to be out of place all the time. What's the equivalent of gravity, if you like, or of, potential energy that is guiding these systems? What is driving this process towards these particular end states? Where is this ultimate goal, the shape of the organism? Where is that encoded? If it's not in the genome, where is it? And
Philip Ball, a huge welcome back to the podcast. I'm super excited to be able to learn from you once again.
Oh, it's a pleasure, Evan. Thanks so much for having me back.
You start How Life Works, which is a truly, truly amazing book, kind of detailing everything about what the best experimental evidence says how biology works, which is radically different, I would guess, to how most people think biology works, and even very different to how I thought biology works. Someone who runs a biology podcast, that's mildly embarrassing, but very different to how I learned how biology works in college and everything. But where you started that book was in in genetics and the gene picture. So maybe lay that foundation. Like what have we gotten wrong about genes in the last couple of decades?
Well, I guess, I started with the traditional idea of how biology works, and I'm going to put it simplistically, but I don't think it's a gross distortion to say that probably since the beginnings of molecular biology and modern genetics, which is, the 1950s, Crick and Watson and DNA, the idea has been that life works because it's a program that's encoded in our genome and is just sort of read out. And it's read out to build us, and then it, continues to be read out as we go about our daily lives. So it's an idea that information in biology and causation actually flows from the bottom up, from the genes upwards. And, that made a lot of sense at the time because the genome is this string of information that's kind of it looked at the time like a string of, computer code like you got on magnetic tape in those days. And I remember those days, the computer code was encoded on magnetic tape. So it looked, as though this was how things were working. And that picture got us a long way in terms of understanding biology as a kind of information science, where there is information that's, it literally is encoded in DNA and is read out to make proteins, and that information is what's inherited. So it's not as though that picture was totally, wrong or fantastical, but it's not enough. And not just that it's not enough, but it gives the wrong impression of where the information is coming from, where the organization in biology is coming from. So, that was the traditional picture. It was kind of encapsulated in Francis Crick's central dogma. I mean, just the fact that it was called that sort of tells you everything. The central dogma of molecular biology, which Crick was quite careful about explaining, was really a statement about how this encoded information in a genetic sequence, how it moves around, how it's used by the cell. Basically, that it's used to, first of all, make this intermediary molecule RNA, and then the RNA molecules go away and are converted by a big protein complex called the ribosome into proteins. And all of that is fine. But the sort of implication was, and I think the way that message has tended to be received, is once you've got proteins, job done, because the proteins are little machines that somehow put us together. And that's not the way it happens. And it's really something that I think has only started to become clear over the past 2 or 3 decades, not coincidentally, since the completion of the Human Genome Project. So that old picture gave the message that all we now have to do is to decode the genome, and we've kind of got it all. We've got the answer to how life works. It hasn't turned out that way. We've decoded the genome. We've decoded now, tens or maybe hundreds of thousands of genomes, and we still don't fully understand how these complex systems of cells and of organisms work. And I think it's become clear not only that the information isn't all going to be found in some encoded form in the genome, but that actually every step of the process is slightly different, or sometimes profoundly different, from what we've learned. So that's really what I set out to do in the book, to step through those processes kind of at ever larger scales in the biological structure of the organism, to explain how things have changed in the past 2 or 3 decades. And even at the level of genes, there's been quite a lot of change.
I love it. I love it. So well explained. Because it's so intuitive to assume that the causality is from the bottom up, right? I think, and we'll get into causality will be a kind of one of those themes that goes throughout. It's just such a weird concept to think that it could be any other way. So what is the right way to think about what's happening there if the information is not coming from the bottom up? If genes do specify proteins, and proteins are machines, and point out where any of this might be incorrect or where more nuance is needed, and then proteins kind of build the rest of the organism. What's wrong about that specifically?
I guess my real answer to what's wrong about that is first of all, that is not what modern molecular biology shows. So it's an empirical answer. And I was really, really keen to make that the guiding principle of the book. I didn't want, I'd seen there have been for, years and years, actually, voices who've been saying, this isn't the right picture, and they've tended to come from a slightly abstract or slightly sort of philosophical position about what organisms are and what biology can be. But I wanted to root it in what we have found out at the lab bench in terms of the way these things actually work. And
Yes.
So I argue that it's that alone that compels us to think of a different kind of narrative. But, when you think about it too, building an organism by that bottom-up way would be a terrible way to do it. It would be an insane way for, let's put it this way, for God to plan us. Because organisms are not like that. They cannot work like machines. If they worked that way, it would require every little bit to have to be in the right place at the right time, like a piece of machinery with billions of cogs, each one of which has to fit in the right place and has to turn. And if one of those is out of place, then the whole thing falls apart and stops working. You're never going to get a complex mechanism to work solely from that way. plus the nature of the biological organism is not the nature of a machine in the sense. So in the following sense, for example, if the idea was that the genome tells the organism everything it has to do in any sort of circumstance, I mean, how is that ever going to work? Every organism, and particularly complex organisms like us, every day encounter circumstances, situations that we have never encountered before and that our ancestors have never encountered before. It would be absurd to imagine that somehow there's a kind of lookup table in the genes for what we
Right.
should do in those circumstances. That is just not going to happen. We are, as all organisms are, we are making use of information from outside all the time. And that information, that context needs to be what is governing our decisions. And so we need to have some way of integrating that information. It's a familiar idea to neuroscientists, that somehow our neural circuitry integrates information and comes to a decision about what we should do. But actually, that same principle applies all the way down the biological hierarchy to the simplest living organisms, to single cells.
Yes.
They too have to be able to make contextual decisions. So it can't all come from the bottom up. There has to be some way, ultimately, I talk about it as there has to be some way to ensure that biological organisms are genuine agents. They're not just automata programmed by their genes.
Getting accurate and unbiased information from the latest scientific research is critical for my job as a science communicator. That's why for over 1 year now, I have been doing all of my scientific research with Consensus.ai. You can think of Consensus as the ChatGPT for science, trained exclusively on research papers, so it doesn't give you that hallucinated garbage like ChatGPT. Recently, I've been in love with the new deep search function they just launched. Let's try it out. So I needed to learn about brain organoids for a podcast with Philip Ball. Let's run a deep search and see how Consensus helps me understand the entire breadth of a field in literally the shortest time possible. It filters through every paper on brain organoids, giving a really nice intro summary at the top. It comes up with this really interesting question: do brain organoids model human brain development accurately, and gives me a consensus bar on the evidence. It tells you the top contributors in different fields. This is a table I love: claims and evidence. It lists out the most relevant claims to your original search and ranks them by evidence. I mean, seriously, where was this while I was in college? I think that's about 20 hours of work right there in one button click. If you're a scientific researcher, a student in college, or you're simply curious and have questions about science, then I think Consensus AI is a complete no-brainer. And the best news is that you can use my link in the description to get a whole month completely free, so there's really no reason at all not to try it out, and it really supports the channel. Thank you so much.
Yeah, really well said. The expression that I love for this is biology is cognition the whole way down. This is sort of Michael Levin's work and Anna Chaunica, and it's just such a beautiful way of understanding biology. And it's just, I think you can't help but not get goosebumps when you read about it. You're like, wow, that's just simply so much more marvelous than I ever could have expected. After finishing this book, what do you think is the right way to think about what genes do? Because I thought that was, you go through a number of different pages and chapters of even just trying to define what a gene is very hard. Because this whole one gene, one function is completely wrong. This one gene, one protein, very wrong. So what is the right way to think about what a gene actually is and what it does?
Yeah, well, that's a discussion that I wanted this book to start. So, I'm not by any means convinced that I have hit on the right way to think about genes, but there are some ways better than others, I think. And it's certainly, I mean, it's not even the idea anymore that the gene is an instruction for how to build a protein. It's an instruction for how to build various proteins on the whole, particularly in our bodies, because the gene, encodes possible proteins of which several might be made, sometimes in different tissues, because we now know, and we've known since the 1970s, that when that information gets translated, get transcribed, I should say, into RNA, into messenger RNA, before it then gets converted into a protein, generally it's chopped up and edited, and bits are thrown away and the other bits are stitched together. And you can do that stitching in various different ways. And it does seem to differ very often in different tissues. So literally a different protein is encoded. So, even at that level, the level of the central dogma, it doesn't mean the central dogma is wrong, but it does mean that it needs to be modified and, made more sophisticated. But I suggest in the book that the way I think about the genome now is that, and I should say the genome rather than genes, because there's not even a clear distinction anymore between which bits of our DNA can be considered genes, and which bits are, just sort of evolutionary junk, of which there absolutely is going to be some, and which bits are something in between. And actually, there's quite a lot that's something in between. I think that the modern view of the genome makes much more fuzzy the distinction between what is functional and what isn't. I kind of think of the genome now as encoding kind of adventitious matter, molecules that might have a function, that don't have a well-defined function, but that might be useful to the cell. Tiny little proteins that are almost too small to be proteins, for example, tiny little stretches of RNA that don't encode proteins but do things themselves, but do them, in combination. And we'll talk about this more. So there's all this stuff that's coming out of the genome. And so I think the best way to think about it is it's a resource. It's a source of resources, of molecular resources that cells can use. So, some of those have well-defined functions. There are some enzymes that are well-defined in terms of their structure and their function. They have to be like this to do their job. The cells absolutely need them to do their job. So, that's quite clear. But there's a whole bunch of other stuff that is much, fuzzier, that we can't even speak about it having a single function. It seems to crop up in all sorts of places, or these molecules might sort of act collectively. So that's the way I think to think about it. It's a source of molecular resources for the cell.
Yeah. It's well defined, but I think it's sort of apparent how language just fails at biology. It's so hard to encapsulate biology in language, because biology is this fuzzy, chaotic, and oftentimes random system of continuums and spectrums, and language is all about labels and discrete chunks, and this is this and this is that, and it just feels such a poor medium of describing biology. Like, we almost need to add another dimension to it. Like it needs like a 3D animation or a world or virtual reality or something to really get at the essence. Because just any linear sentence you use to describe biological processes, you can always point holes in and say, well, actually, here's an exception here, and it doesn't really work like that in this case. And just, you can't really get that fuzziness. And it's why I appreciate how well a job you do at it in writing, because it's just so difficult. I mean, have you found that you thought that just the sentences or just the language just doesn't really seem to get at it sometimes?
Yeah, absolutely that. And again, that's why I wanted the book to start a discussion, or maybe it's already happening, to contribute to the discussion of what narratives we need to describe biology now. If we recognize, as I think we should, that the old sort of computational linear narrative is no longer the right one, how else do we talk about this? Because we absolutely need narratives to understand biology, or to get some foothold on it. None of them is going to be perfect, but we need them to do some kind of job. So yeah, it's certainly true that language is a problem, but I think what has tended to happen in the past is that, for that reason, and also because this is sort of a way that our minds work, we have fallen back on metaphors and analogies from our technologies. So, and this is something that's always happened for the body as a whole. I mean, this goes right back to the sort of mechanistic philosophy of Descartes, and in the 17th century, that he thought of the body as a machine, and we have continually thought about it as a machine. But the nature of that machine has changed, whether it's sort of hydraulic, or whether it's computational now, or whether it's mechanical. We talk about it in machine terms. And the one thing I really wanted to stress in the book is that isn't going to do the job in biology. There are occasions, there are some proteins, for example, that really do look like machines, and we can use machine-like terms to understand them. There are proteins that look like sort of rotary motors, and we can think about them in terms of torque and,
Yes.
those sorts of concepts. And that's fine. Most proteins aren't like that. We talk about them as molecular machines, but actually they don't behave anything like our machines do. They're much too fluid and stochastic and all the rest of it. But the body as a whole is nothing like a machine. We don't, machines tend to be made from parts that have particular functions, and they're there to do a job. And again, that's not in general the case in biology. It's sometimes the case, but some of the most important bits of us at the molecular scale and upwards aren't bits that we can say, oh, this protein does this. It's this sort of protein. We find them popping up in several or maybe many different contexts. So, what they do depends on their context, and that isn't a principle that we generally find in machines. So whatever language we use, I think we need to drag it away from this idea that, we talk about it in terms of mechanism and machine.
Yeah. Yeah, I think the animations that we do have as well definitely paint the proteins in that machinery-like way. Like you see the flagella of the bacterium, which literally looks like it was constructed in an engineer's lab and just like plugged onto a bacterium, which just is this big motor. And like the ATPase, which is like cranking different enzymes together. And they really do look machine-like in the way that we've animated it together, because again, it's just like extremely hard to do it any other way. I guess it's hard. You can't encapsulate that fuzziness and randomness in, and the dynesin, the kinase, when they're dragging molecules down actin filaments, and like all these animations that we have are just so machine-like because it's just so hard to do it any other way.
Yeah, it is, and I understand that. And a lot of those animations are ones of molecules of proteins that are more machine-like. So
Yeah.
these motor proteins, they are a little bit, we call them motor proteins. There's a metaphor, and it's not an unreasonable one.
Yeah.
But, many proteins aren't working that way at all. And those animations, I totally understand why you have to simplify them, why you have to do something like this. But it does make me smile when you see proteins kind of zooming in like little,
Homing beacons
hovercrafts or drones or something, guided to their target,
Yes.
as though there's some force that's sort of pulling them there. And that is not, how the molecular world works. Of course, it's totally diffusional. And not only that, you see them as though this is happening in some big open space where things can fly around. Whereas, we now have good snapshots of what the cells really look like, and it's chaos in that.
Yes.
They're absolutely jammed with molecules sort of, bumping into one another. So you couldn't possibly do an animation that would make sense of a picture like that. So, I understand that, but it does give us the wrong impression of not only how these things work, but what is the nature of the kind of process, we're talking about, which cannot be machine-like in a place that is so stochastic, so noisy as living things are.
Yeah. Are genes selfish?
I think that, there's a section in the book that critiques the notion of a selfish gene, but I wanted to come at it from the point of view of thinking, well, look, this has been a useful way for some biologists to think about what genes do. So let's not throw the baby out with the bathwater. And the way I kind of think about it now is calling genes selfish is not it's saying anything about the nature of genes themselves, nothing whatsoever. Obviously it's metaphorical, but even as a metaphor, it doesn't work for what genes actually do day to day in our bodies. It's a metaphor for describing the model that many evolutionary population geneticists use to think about genes. So that's really what it is. It's not it's not a metaphor about genes themselves or about reality. It's a metaphor to capture that particular model, which is a model that can be useful if we're wanting to understand how different variants of genes spread through a population. And we can do that without any reference to the things that are actually alive, living organisms. In fact, when we do that in those models, the organisms become a puzzle. They become what Richard Dawkins called, there's a paradox of the organism in that model, because you start to think, well, why are organisms needed at all? And the best you can do, and this is what Richard has famously talked about, is to say, well, then the organism is just a vehicle, just a passive vehicle for the genes, and it's the genes that are calling all the shots.
Yes.
In that model, that is how you think about it. If you then make the mistake of thinking you are talking about actual living things, about how biology actually works, at the molecular scale or at the organismal scale, then you've lost sight of what you're actually talking about. You've lost sight of the fact that you're talking about the model. So if genes are selfish at all, let's confine that to that particular arena of evolutionary population genetics. There is no meaningful way in which the way genes function in living systems can be considered selfish. There are actually bits of our genome that I think you could reasonably describe as selfish. These are things like transposons, these elements that are just exist to kind of make copies of themselves within the genome. And if they were allowed to do that, if they were given free rein to do that, they're often sort of bits of viruses that have, made their way into our genome. And if they were allowed to do that unchecked, they genuinely would be selfish in the sense, because this is really what selfishness is about. It's not just about looking out for yourself. It's looking out for yourself at the expense of everyone else.
Yeah.
And if they were allowed to do that unchecked, the genome would kind of fall apart pretty quickly. It would just stop working. And that is precisely why we have very sophisticated molecular sort of immune systems within the cell nucleus to keep those transposons under control. So the notion of there being selfish bits of DNA is a perfectly good one. And in fact, it was one that predates the Richard Dawkins selfish gene. That's another of the worries I have about it, because there was a perfectly good, sensible way to talk about selfish DNA that became muddied once you said, well, actually all genes are like this.
Yeah.
So, we just need to be very clear what we're talking about when we talk about things like selfish genes.
For sure. And I think a consistent theme throughout this conversation will be metaphors have their place. They do some things well, but there's almost always exceptions in biology. Like there are some applicable metaphors. No metaphor is right or wrong. They're just useful, I guess. And some of them are useful in some places. And then when you try and draw too wide boundaries, you're just always going to find exceptions. Because again, biology is not neatly packaged up into any sentence that would, unless it's just so broad and kind of meaningless in explanatory value, you find exceptions to everything.
Yeah. No, I absolutely think that's true. That we, first of all, we do need metaphors, just like we need narratives. That's how our brain works in making sense of the world all the time. Language is full of metaphors. Language always consists of metaphors. So, it's not a question of doing away with them. But the problem is that we don't have any good mechanisms in science for kind of constantly reevaluating them and thinking, well, does this metaphor still is it still fit for purpose? And so they just kind of stay. And often they stay and they become what are called dead metaphors, which doesn't mean ones that aren't used anymore. It means ones that we've forgotten that they're a metaphor in the first place. And I think one of the problems I have also with the selfish gene metaphor is that it can become, metaphors are meant to help us think, but actually it can be required to do explanatory work. And then that becomes a problem. If the metaphor itself, we need genes to act sort of selfishly as though they're little agents for that model to work. They're not agents. They cannot be agents. They do not have what is required to be agents. But we need, for the selfish gene model to really be a description of reality, we need to turn those genes into little organisms. And all the agency of the organism itself has been taken out of the where it actually belongs, where it actually resides, and has been put into the genes. And, unless we sort of see them as selfish, we can say as much as we like, oh, it's only a metaphor. But actually, in that case, I think the metaphor is required to do actual work in making it, happen. And that's a problem.
Yes. Another really good point. You have a quote in the book we will never have adequately explained life until we understand where its robustness comes from. Explain why robustness is such a critical feature of life.
It's again, it comes down to this idea that we are incredibly complex, and the more we look into the molecular kind of mechanisms of biology, the more you kind of put your head in your hands and think, how can we make any sense of this? And yet here we are, after, don't know, in your case, maybe 30 years or plus, in mine, considerably more than that, and we keep going. So, it's extraordinary. The more you kind of look into it, the more extraordinary it seems that we work at all, that we have this robustness. Which, of course, we have to have, because all organisms face uncertainty. They face uncertain circumstances. You don't know what's going to come. And so there has to be some kind of leeway for, dealing with that. And things are going to go wrong all the time, and things do go wrong all the time within us, for sometimes worse, sometimes not so bad, but one way or another, we find organisms find a way through. They also, during development, as we grow from an embryo, it's not a kind of, total sort of painting by numbers or sort of Lego assembly where everything is going to be definitely put in the right place. It's kind of organizing itself, and there are going to be, errors sometimes there that have to be put right or have to be accommodated in some way. So that's what living things have to be able to deal with. And the question is where this robustness comes from. And I don't think there's a general answer to that. But there are various ways that people have sort of talked about that. For example, when we're thinking of what a particular gene or gene product does, all our genes there's variety among them. They all have mutations. We're accumulating mutations all the time. Sooner or later, there might well be a mutation that stops that gene or the gene product from doing its job properly. If, as soon as that happens, as soon as you get a sort of faulty protein made somewhere, everything goes to pot, then, that's not going to give you any kind of robustness. So how do we sort of buffer ourselves against that? And a common way of thinking about it in the past is to say, well, there are these networks of genes that sort of and proteins that interact with one another, and there's always different pathways. So if this pathway stops working, there's another one here that can get to the same place. There's something in that, because we can see situations where that happens, but I'm not at all persuaded that in general is the best way to create robustness, because in a sense what it's saying is, to make a complex system robust, you've got to make it more complex. You've got to build these backup pathways that might never be used. And it just doesn't seem sort of logically to be the best way of doing it. And I think that that modern molecular biology is showing us that robustness comes much more from making sure that whatever happens at the lower levels of the system, actually the fine details don't matter too much to what happens at a higher level. So, the functioning of one of our organs in general isn't going to require every single cell to be working properly. And the functioning of those cells isn't going to require every single protein or other molecule to be in the right place at the right time, because that's not going to happen. So living systems seem to build in at every level a kind of fuzziness, a kind of leeway, I suppose, so that those fine details aren't going to derail everything. And that seems to be a more general principle than just having a backup plan.
I'm trying to think of what the right, I'm again going to metaphors for everything, but I'm trying to think of what the right factory metaphor for this that actually works. It's like there's loads of conveyor belts, but the conveyor belts are constantly shifting and changing their parts amongst themselves as well as going linearly. So they're like non-linear conveyor belts that are constantly exchanging parts and shifting parts with each other. Each on average maybe have a function, but each can also produce any other part of the pathway. And really the sort of a core function of the network is that they are just constantly exchanging parts with each other and on average they might do something, but they're really promiscuous, and they probably all have the capability to do all of them, or they have a wide range of function. Is that maybe, again, metaphors are flawed, but is that sort of the way you can think about it?
Well, I think what that shows, Evan, is that when we fall back on traditional machinery metaphors quite quickly, they don't work, and we have to start imagining some quite fantastical machines.
Exactly, yeah.
I think a better way of thinking, but I find it more useful to think about it in more sort of sociological terms, of how people work, for example. So to take an example of how our genes are regulated, how they're switched on and off. The traditional view of this, and it's something that seems to happen in bacteria and other single-celled organisms much more, is that there are sort of little switches that a molecule from one gene might come along, a protein might come along and stick next to another gene and turn it on or turn it off. So it's very like little digital switches. For us, that's in general not how it happens. There's a whole bunch of molecules involved in regulating every gene. There are these proteins called transcription factors, but they behave, not individually. They behave in pairs or in groups in a context-dependent manner. There are other molecules, including non-coding RNA molecules and other bits of DNA from elsewhere on the chromosome that kind of come in on big loops to sort of switch on and off. It looks like an incredible mess, but I think of it as more like a committee of molecules that come together to make a decision. Should we turn this gene on or off? That's what they've got to decide. And, our committees, they work and they're sort of planned on the assumption that sometimes people are going to be sick. They're not going to be there. Or, sometimes people just aren't going to be listening, or they're going to be falling asleep or something. Not everyone's going to do their job. And that doesn't matter too much. As long as the committee is quorate, then it can come to a decision of some sort, hopefully, the right decision or a reasonable decision. So I think of these, and, this is an example of many processes in cells. They're committees of molecules of many different molecular components that interact more loosely, and that somehow, in a way we don't understand at the moment. We know a few things about it, but we're not totally ignorant, but we don't understand the principles. But somehow, on the whole, they seem to come to reliable decisions in the end. So, this is how our societies function. This is how we run a company on the same basis that, that some of your employees aren't going to be there. They're going to be on holiday. They're going to be off sick. And so someone else can kind of step in and compensate. So there are these kind of compensating processes that might happen amongst the individuals, or it might happen at a higher level of organization that makes up for all these fluctuations at the lowest level.
Yeah. Like, do you think there's obvious hierarchies in biology, and an obvious hierarchy to think about it is like organs or humans, organs, cell aggregates, all the way down to much smaller biological compartments, proteins, all the rest. But do you think there's the way you said a committee there, when you have all these different pathways, do you think there's even micro hierarchies that almost like every stage of the hierarchy isn't just one stage, but is also a hierarchy of stages where in that committee, there is some sense of some parts being more important, or some parts orchestrating those processes a bit more than others. Some parts are more promiscuous than other parts. You know what I mean? That there's even hierarchies within these biological systems.
Yeah, I think absolutely. And that's really the core of the essence of the problem we're trying to understand, that it's not just a bunch of, random molecules coming together and doing something. Those molecules are there, they have particular purposes to fulfill when they're there, and they're not all of equal importance. So, transcription factors, there are lots of different places that a given transcription factor will stick, and often they might stick, or they might stick to the same place and do a different job, or they might stick to a different place in different cell types, so in different tissue types. But it's not random. There is some kind of logic to it. So that's the challenging thing to understand. And I think what's interesting about living systems is that this kind of separation into hierarchical layers of which each only sort of depends quite sort of loosely or in a kind of coarse-grained way on all the details that went on below. That's sort of generally the way it happens and needs to happen. But it's kind of leaky in the sense that, for most of our genes, for example, as I said, they do acquire mutations, and it doesn't matter for some reason or another. Somewhere along that chain, it takes care. It's sort of smoothed away. But sometimes it really does matter. Sometimes, a mutation could be fatal, or will be carcinogenic. And sometimes, particular gene variants in the population are potentially lethal. Cystic fibrosis, one used to be sort of monogenic diseases, generally are sort of well known for that. So sometimes what's going on at the lowest level really does matter all the way up. But most of the time it doesn't. And I kind of feel that this too makes kind of intuitive sense when you think about it, that sometimes, biology has to be able to heed what's going on down at that lowest level. It needs to be, there needs to be some channel of communication. But, on the whole, there's so much kind of variation down at the smallest scales that on the whole, biology needs to find ways of kind of integrating that away and still being able to work. So it's this very sort of delicate balance. And again, you can see, you can sort of see this in society too, I guess, that there are, some people in a company who, if they're away, it's much more important than if others are away. So, there are metaphors like that, but it's harder to think of any process like that in our standard sort of machine thinking.
Yeah. Every part is critical to just the overall function. There's no excess parts you highlight really well, I think, these gene knockout experiments where, scientists might have thought that they isolated some gene that was critical to the developmental pathway. They knock out that gene in a mutant, and then the thing just develops normally. And you're like, well, that's weird. But I think that's sort of explained by this, in that there's multiple ways of doing everything. Because it hasn't been overly, or there's very few, I would say, individual genes, as we pointed out, like cystic fibrosis, Huntington's, that are so critical that there isn't promiscuous ways of doing it, which is
I think that's right. And, sometimes a knockout like that, the traditional way of talking about it was in terms of that sort of redundancy of pathways. So, if the gene is knocked out, well, there's another pathway that can make up for it. Sometimes that's true, but I think probably more often what's happening is there's some way of the higher level of the system is able to compensate for the absence of that gene. And that's why it's wrong to think of causation totally flowing from the bottom up. That causation actually happens at a higher level, and it was those kinds of experiments, those knockout experiments, that started me actually decades ago, kind of thinking, hang on a minute, are we really talking about this in the right way? Because it was when I was an editor at Nature, and I was an editor of physical sciences, but we'd have these weekly editorial meetings, and my biological colleagues would come along, and they'd often be saying, oh, we've just accepted this paper, it's a knockout paper where we thought this gene was involved in such and such, but it turns out, there's this knockout experiment that either that doesn't make any difference if you knock it out to that process at all, or that we find that it's actually really important for this other process as well, which seems totally unrelated. And I remember, one week I finally built up the courage to say, so what does that mean? And the response was, very frank, we have no idea. We don't know.
Yeah.
And I suppose it made me start to think, well, are we telling the right story about genetic causation if we keep finding this happen again and again? And that's really, I think, the position we're at now. That I don't think it was, that sort of linear picture of causation wasn't the right one to understand these knockout experiments.
Yeah.
MindLab Pro is a scientifically backed nootropic designed to enhance memory and focus and keep you as mentally sharp as possible in 2026. And before you click off, it has been scientifically proven in not one, but 3 separate double-blind placebo-controlled trials that it really works. In one study, healthy adults took MindLab Pro for 30 days and improved on 3 separate information processing tasks. In another trial, adults improved on a set of memory tasks. The interesting part is in both trials, this improvement did not happen in the placebo group, which is a really strong piece of evidence that what is making the difference is in this bottle. MindLab Pro has no proprietary blends, complete transparency on ingredients, and has full third-party testing so you know exactly what's in the bottle. Performance Labs is one of the most scientifically rigorous supplement companies that exists, and that is the exact reason that I have decided to partner with them. I also take their sleep stack every single night, and they sell a bunch of other high quality supplements. They offer a 30-day money back guarantee, so there's literally zero risk in trying them out. You're supporting the giant shoulder by using the link in the description. Now back to the episode.
What's a really good example of understanding what cells are noisy, or the activity of a cell is noisy? Like, what advantage does that give the cell? Is there a good example that you have for like why would the activity of a cell be noisy?
Okay, so yeah, that means that any molecular system, I suppose to a chemist, it sort of makes sense that this is, the case, that we chemists kind of think of, and I trained first as a chemist, we know that chemistry is noisy, that it's stochastic, that things are kind of moving about, with Brownian motion. Whereas, it seemed to me that the biological picture, the old picture, was much more deterministic than that. It actually seemed to deny that stochastic, noisy character of chemical systems, and seemed to be implying that, everything was like those animations where everything is kind of moving into the right place. So there is this noise that, is intrinsic to the molecular world, and biological systems have to be able to cope with that. And, we've talked about some ways in which they're robust against that. But I think it totally stands to reason that actually sometimes biology has found ways of using noisiness. And, I suppose one example of that is just the way biology can, noise is a way of trying things out. So in and not getting stuck in the wrong states.
Yes.
So, it's a noise is made use of for classic sort of computational algorithms for optimizing things. There's one called Monte Carlo algorithm, which is, a way of finding the sort of optimal outcome by sort of trying things out bit by bit. And, the way that's kind of done. I'm really describing the process called simulated annealing, where you kind of slowly sort of cool the system down and hope that it's going to converge into its most stable ground state. But you don't want to get stuck in other states along the way that aren't the lowest energy one. And so you make sure there's a bit of noise in there so that it can continue to explore locally and not get stuck in little, local minima, but can gradually find its way down. It's the same process as why we shake the salt cellar, to add a bit of noise so that the salt comes out and doesn't get stuck in some jammed state. So biology can, make use of that too. And I think we see that in developmental systems where, the challenge for a growing embryo, growing into a complex organism like us, is for all the cells to develop into the right kind of cell type, whether it's a heart muscle cell or, neural cells or skin cells or whatever, in the right place at the right time. And that's going to be very noisy. And we can actually see now, because we can track individual cells during development, and we can sort of see them almost exploring the landscape sometimes. And sometimes, they're going as though towards a neural cell fate, for example. And then because of the signals they're getting from cells around them, they kind of realized, to put it that way, that's not the right thing to be at that time in that place. And so they change their mind and they sort of go somewhere else. And you need that noise to have that sort of adaptability, that
Yes.
constant, adaptability. Whereas if it was just following a program, an inevitable program to become a nerve cell, then things are going to get screwed up and things are going to be out of place all the time.
Yeah.
So yeah, that noisiness is a way of finding your way to the right place in the landscape and not getting stuck in places you shouldn't be.
Yeah. Like, I think we underestimate the fact that every cell in our body, despite a few examples like sex cells or red blood cells, have the same packaging of DNA, but still our cells the DNA in our kidney expressed kidney cells, neural cells, pancreas cells, liver cells. Like, that's a phenomenally just we just don't, I think, appreciate that fully. And you're right that I think without the noise, you would just end up with like a Lego character, but different arms, different legs, different, you know what I mean? So all the different pieces would just be jumbled up and it wouldn't be able to know. You have a really good example, I think, in the book where you talk about hematopoietic stem cells in mice, ones that decide to become either red blood cells or white blood cells. The fate decision is largely controlled by two transcription factors, GATA1, PU1, with another player, GATA2, in the mix. And in the biology textbook picture, what you'd imagine is like this tiny genetic toggle. Flip one way, become red blood cell. Flip the other, become white blood cell. But what Steidl and colleagues actually found is that inside real cells, the genes for GATA1, GATA2, and PU1 fire in these sporadic random bursts of transcription. And they found that using fluorescent tags on the RNAs. And they literally saw the cells blinking with noisy pulses of all three, which is just phenomenally beautiful start, even though these factors are meant to be antagonistic to the system. So it looks like chaos, but then when they modeled the system, they realized that the noise actually helped, that those transcriptional fluctuations made it easier for the cells to reliably reach either of the target cell lineages instead of getting stuck in this halfway spot. Noise keeps the fate options open, lets the system explore the landscape, and then tips it into a stable identity. When I read that, I was like, that's so cool. Amazing.
It really is. And thank you, Evan, for getting that very specific example of the kind of general thing I was talking about there. I mean, that's an indication of how, in this book, I've gone into the weeds sometimes.
Yes.
I felt like I've really had to show this is real stuff. This isn't just some metaphorical idea about what cells might do. We see this happening in very particular cases, and there's one. And, that's a reflection of, so what you have in a situation like that is a sort of delicate balance, really, that you have, there are what we now talk about and think of as attractors in that landscape of possible states the cell could have, a massive landscape, the number of different genes, combinations of genes that could be on and off in any cell. I mean, it's literally astronomical. And yet somehow, they find their way into a finite set of cell types. And we don't know how many, because we don't quite know how to define a cell type. But, we can meaningfully think about a skin cell being different from a, nerve cell or something. And they find their way into that because that huge dimensional space is somehow kind of condensed by the the biological process into a much lower dimensional landscape with attractors with different cell types in. And the question is how the cells navigate between these attractor states, and that can be described. I talk about it in the book. It can be described using the language of dynamical systems generally, because we're familiar with a sort of complex double pendulum or something. We'll have different attractor states, or the solar system has different attractor states of where the planets orbit. So we're familiar with it from simpler systems, but it seems the same ideas apply in biology, and it makes perfect sense that there would be this landscape of possible states with quite sort of broad basins. You don't have to have exactly these genes on and these genes off, but you have to be in that right sort of vicinity to be that kind of cell type.
Yes.
And there's this balance between that's a kind of deterministic picture where you're drawn into particular attractor types, but also the stochastic picture where you can be exploring the landscape to find them. And what we've now known for sort of 20 years that actually you can tweak cells in what seem to be quite small ways to make them jump from one attractor to another. And in particular, you can make them jump from the attractor of, say, skin cells all the way back to the big, broad attractor of stem cell states, where cells that can develop into any tissue type. And you can do that remarkably easily just by treating cells with 4 or 5 different factors, basically proteins or gene products, to get them to make that sort of journey in the landscape back to a different type. And so this way of thinking helps us to understand how it is that we can reprogram cells to get them to develop into particular tissue types, maybe for regenerative medicine, maybe even for treating cancers.
Yeah. I love these metaphors. I think it's one of the best metaphors that biology has, is this dynamical landscapes of valleys and forks. It's just such a beautiful way, I think, of really conceptualizing what the molecules are doing, what the cells are doing, what the systems are even doing. It's all sort of these dynamical landscapes. Can you kind of explain a little bit more on that? What is this idea of canalization?
So this picture, this landscape picture, goes back to the developmental biologist who called himself an embryologist back then, Conrad Waddington, in the 1940s. And he famously drew this picture of it's like a sort of system. It's like a river drainage network, a system of sort of valleys that where the cell types, but it's sort of like a drainage network in reverse, where you start off with one valley and it splits into 2 and then splits into 4 as the further down you go, to represent this notion that you start off with a single cell state, stem cells, and then they split into maybe 2 or 3 different cell lineages, each of which can become all sorts of different types of tissue. And they progressively specialize until they reach the mature tissue types, as cell types. So that was Waddington's idea of a developmental landscape. And it was a very influential idea, but it was kind of seen as metaphorical until we got to this modern view of being able to actually see which genes are on and which genes are off in individual cells. And we can actually literally now start to plot out what these landscapes look like that are, basically the real versions of Waddington's sort of metaphorical ones. So we can actually see what this landscape looks like. And it's more complex than Waddington's. For one thing, you can find some valleys kind of have loops that, come round to the same sort of end state. They're not just continually branching, but, we can actually draw physical pictures of what this looks like. And canalization was a kind of part of that, in that, as the sort of word implies, really, that these valleys, they're not narrow, they're quite broad. So you can start from various positions and still find your way, into the same valley, so that the number of choices that cells have is quite restricted. They can potentially be in these different states, but they're still in the same broad valley, and so they'll still be heading in the same direction and end up the same way. And that canalization is a way of, again, dealing with the randomness and the variability that we'll find at the very sort of lowest levels of the system, that you can have some noise and some variation, but you've still basically got the same kind of cell type. Yeah. And I think that's an idea that kind of applies more broadly, and not coincidentally, in biology, in the sense that, I kind of think of it, for example, in terms of disease, that actually it's very striking when we think about it, that many different conditions manifest with similar symptoms. That was one of the problems during the pandemic, that we, found it very hard to know if we had COVID or if we just had cold or a flu. They manifested in, there was a lot of overlap with the symptoms. And that too, I think, is that there's a physiological canalization in the body, where different processes at the molecular and the genetic scale can have different physiological or phenotypic expressions. There too you get a kind of robustness, that you're going to get the same outcomes, the same sort of stable and generally kind of desirable outcomes from different starting points.
Yeah. So the idea is, with something like 37 trillion cells or something in each of our bodies, that there's just so many possible forms that those cells can be in, but humans and organisms pretty reliably end up in these stable forms, because effectively what they're doing is rolling down an energy landscape. So like a big ball, or any ball will roll down a hill due to gravity, there's energy landscapes that mean it's more energetically what would you say, stable for the molecule just to be rolling down these energy landscapes and falling into these local minimas, which are physiological states like a cell lineage or any. But this I think this sort of extrapolates to all biological processes, right? And not just cell differentiation. That you can think of pretty much all biological processes as rolling down energy landscapes, finding stable energetic configurations for a little while until it's more energetically stable to roll down another hill and roll down another hill, and you're finding all these pockets. Is that sort of how you conceptualize biology and all of the different processes going on?
Yes, it is. I think we can see that this kind of canalization at many levels. So, for example, if we think about development again, there are certain stages that the embryo, certain sort of morphologies that the embryo has to go through. And what the most crucial of which, I guess, is often talked about is gastrulation, where there's a kind of I suppose I kind of think of it as the moment when it stops looking like a ball of cells and starts to look like a little kind of embryo, that there's sort of a particular, tissue folding that goes on then. And again, cells have to get to that state. Embryos have to get to that state somehow, even if they're starting from, slightly different places and the cells won't all be in identical positions. The question often is, you talked about this being balls rolling down a hill, because they're sort of minimizing their potential energy. The question is often, well, what is the equivalent in biology? Are they minimizing it? What are they minimizing? Because we are systems that are inherently out of equilibrium, and we're dynamical systems. So we're not looking to get to our lowest energy state. If we were, that's being dead, basically. So, what is it what's the equivalent of gravity, if you like, or of, potential energy that is guiding these systems? What is driving this process towards these particular end states? And I don't think we have a very clear way of thinking, of conceptualizing that problem, of thinking about, biology that way. If we think about it in terms of dynamical systems, then, we're thinking about it as an energy landscape, but it's not that. And it's what it comes down to is that there seem to be goals in biology. There seem to be things to aim for, and in the developmental process, the thing to aim for is the fully formed, properly formed organism. What is guiding that process at each step is still, a little bit hazy. We kind of think of each bit of the process in its own terms, but why it all sort of ends up, where is this ultimate goal, the shape of the organism, where is that encoded? If it's not in the genome, where is it? And I don't think we quite have the right story to tell about that at the moment.
Interesting. Maybe I had a misconception there. So what I had in my head is that a molecule or anything rolling down a hill in biology is moving from a less stable to a more stable state, basically electronically more stable state. There's that saying that science is just electrons looking for a place to rest. It's just more stable configurations of energy. Do you think that is incomplete? Is that not the whole picture? Is there something missing from that?
Yeah, there is something missing from that, because that is what tends to happen, as I say, in equilibrium systems. That they, or systems tend towards an equilibrium state, or
Yes.
energy state. Living systems don't. We maintain ourselves out of equilibrium. So we are these dynamical non-equilibrium systems where there's a constant through flux of energy and of matter, actually. And with that process, even for non-living non-equilibrium systems, things like hurricanes, for example, we don't fully understand that process of how systems out of equilibrium somehow organize themselves into these particular and these coherent structures. For living systems, as a whole, that's even more complicated, because it seems that there is this balance, or this interplay, or this dance between spontaneous self-organization and some kind of information that is encoded in the genome. And we can see that, for example, a great example of that, in us is our fingers, is the development of digits, where clearly there's a genetic element to it, but you will never find in the genome the kind of code for 5 fingers.
Yeah.
It simply isn't there, because we understand this process isn't of that nature. It's not programmed in there. Our fingers are actually, they're basically stripes. They look like that, but they literally are. They are formed, it now seems, through the interplay of a small set of developmental proteins, and they are proteins that aren't about making fingers. They're proteins that crop up in many processes during the developmental process, but they're involved in the formation of digits, and they interact in a non-equilibrium way that gives rise to a spatial pattern. And they do that in a way that was first described in 1952 by the British mathematician Alan Turing. Just sort of hypothetically, he was thinking about how embryos grow, but it was a very, very abstract model. But we now can see that the basis of that model seems to be the way these proteins interact, that they spontaneously organize themselves into a series of stripes. But they're stripes that sort of radiate from the sort of central paddle of the developing limb. And, where there's stripes of one particular protein, they may trigger bone formation, and you get the fingers developing. And in the bits in between, the cells are sort of respond by kind of dying off. And, that's what gives rise to our fingers. So that's a non-equilibrium self-organizing process that doesn't depend, it's not somehow encoded in the molecules that take part, but nevertheless, those molecules are gene products. And sometimes changes to the genes or to the regulation of those genes can cause things like polydactyly, where you get more than 5 fingers. And, I think of that as a kind of metaphor for living systems as a whole. It's this interplay between spontaneous self-organization in a non-equilibrium system, which is something we also see in the non-living world, and the way that is guided, I suppose, by at the genetic level and at higher levels.
Yes, really well explained. I guess one element that I forgot to mention to mine was that molecular processes are rolling down energy landscapes in non-equilibrium systems, but never actually reaching equilibrium. And that constant dance and interplay of non-equilibrium is sort of what life is able to maintain. Do you think one way we could think about the genome is that it's what sets the dynamic landscape? And that's why that's more interesting than saying it's a blueprint, because a blueprint is like a static readout of instructions. But saying the genome is actually this dynamic, constantly shifting landscape where agents are playing out on it. Is that sort of maybe a more useful way of thinking about it?
I think it's a better way of thinking about it. And in fact, that's how Waddington originally conceptualized his landscape, that he drew another picture where the landscape was a bit like a kind of a circus tent or something, or the roof of a circus tent with all these sort of ropes attached to it. And the ropes were attached to genes, and the genes were pulling, on this sort of surface to alter its shape. So he certainly conceptualized it that way. I think that's a fair picture, with the proviso that it's more dynamic than that. The genes aren't fully specifying that. It's the interaction between the higher levels and the lower levels that give rise to it. So, for example, it seems that the landscape can depend very much on, not necessarily on the genes themselves, but on the timing at which genes are expressed. And that just altering that timing, having identical genes, but just altering the timings at which they switch on and off during development, can have profound changes for the organism. And those timings are themselves, there's a whole set of things that they're responding to. They might be waiting for cues from other genes that have to do something first. They might be waiting for environmental signals from other cells or from their surroundings, or even from the womb itself to begin with, in order to switch on and off. So there's still this openness in the informational system. It's not all being determined by the genes. It's being determined by the way the genes are interacting with the larger scale, with the environment and the larger scale system.
Yeah. So the dynamical landscape is established by the interactions of just literally all of the biological components interacting with each other, up and down the hierarchy, sideways, up promiscuously in every direction.
This is the problem. It's so hard to find, language, because it's like, yes, everything. I mean, I mentioned the book somewhere. I remember hearing it in Nature. I think it was Miranda Richardson, the biology editor at that time, said the answer in biology is always yes. It's because everything is happening at once, but it's not just happening kind of randomly. There is a kind of logic to it. And, in a way, because of that, the landscape is kind of shaping itself. That it's, that this landscape, it's not some pre-existing thing. It's kind of moving all the time, so that at a certain stage in development, certain cell types are possible. At a later stage, it's no longer possible to have those cell types. The landscape has moved on, and you have different attractors with more mature cell types.
Yeah. It's so funny to me how different all of this is to what I learned in college and what is even the mainstream way of thinking about biology. Like, I think especially in developmental biology, what I learned was really like a mechanistic, here's the ingredient list, like you're baking a cake type approach, where there was gene gradients and it was definitely complex, but there wasn't that randomness, fuzziness, noisy element in that at all. It was just very much, at this stage, these genes are expressed. It causes this, which then these genes are expressed, and it causes it. And it really was like you're baking a lemon tart. And there's just like, I feel like this intrinsic noisiness is not just a small thing that you can gloss over. It's like fundamental to really understanding what's happening in biology.
Yeah, I think so. I think, the difference between that and baking a lemon tart is, if you realize that you, I don't know, forgot to, add the corn flour to the, lemon after you've baked it, you're stuffed. Sometimes in biology, it can self-correct.
Yeah.
For oversights like that. So it's much more responsive to what's going on. It's not, it doesn't have to be literally one thing after another. At other times, of course, it can be catastrophic if something fails to happen. I mean, I think, for example, the closure of the neural tube is a great example. That the neural tube is a structure in the living system that has to sort of enclose the ultimately the sort of nerves of the spinal column, and the failure of that system, the tissue sort of forms into a layer and starts to curve, and it's got to close. And if it doesn't close, then you have problems like spina bifida. That's literally what spina bifida means. And you can have all sorts of other developmental problems. And it's a situation where sometimes, if there's some sort of problem like that during development, things can happen at a later stage to compensate. And, it's not necessarily catastrophic, but sometimes it is catastrophic. And, this is why it's such a sort of complex condition. And, there are all different sort of variations of how this can happen. So I guess, I think the way to think about development is it has a degree of self-correction. But it can only go so far. And again, the question for me that I don't think we really yet know how to think about properly is we talk about sort of self-correction, but what is guiding that? Where is the
Yeah.
plan? The sort of structure that these systems are sort of aiming for. And yeah, it's really not clear how best to think about that.
Yeah. Yeah. It's probably one of the most fundamental questions in biology, I guess, is like, what is really the driving force behind any of this? I think that's why the selfish gene metaphor was so interesting, or it was so useful for people, is that it's just so easy to understand all of life under that way. Okay, genes just want to replicate. I know there's so many problems with that picture, but that's a narrative that people can understand, and it sort of gives some context to almost all of life's processes. If that's all it is genes replicating, then that explains the whole thing. Now, again, as we said, that's so many problems with that, but like, it's a simple idea and sort of explains it, but there is a fundamental issue there. We don't know what's at the very, very, very heart of it. And it's probably, would you agree that maybe that fundamental heart of it is like a physics thing? Is it not even a biology thing anymore? At the very heart of channeling the entire system, is that like the most fundamental aspect of physics sort of question?
I think it requires what I think of as physics-like thinking to frame the problem that way. But I don't think that it's the concepts that physicists deal with are necessarily ones that are going to give the answer. So I want to be very careful about that kind of distinction, because there's a long and ignoble history of physicists coming into biology and saying we're going to solve all your problems, and they do it in such a simplistic or such an abstract way that it doesn't mean anything to anyone and doesn't really explain anything. But I think it's a physics type question that biologists probably, I think, too rarely, ask themselves: what is guiding this process? What is driving it forward? If it's not, and I think that was really why I think the narrative of the genetic blueprint took hold, because it was a way, in retrospect, it was a way of kind of not having to worry about that question, because we can just pretend it's in a genetic blueprint, which we can no longer do because we can see that it's not. And so we have to, to come back to that question. But I think that it's the same with the selfish gene, that if we can say, well, genes just want to, replicate, and so they're going to build the organism. That's precisely an example of where I think the metaphor is doing work, that you're actually saying, well, then of course it's going to build, because the genes are going to make sure. How are they going to make sure of that? What motive power do they have to do that? It simply does not work that way. It does not give you a proximal answer to the question of how an embryo grows, to say, well, it's because the genes want it to, because they've got to have this vehicle. So it's giving genes agency in that problem that they don't possess, and so we've got to go back and think, well, how is that process really happening?
Yeah. Let's go to causal emergence. I think this concept is super interesting. First off, let's explain that idea. What does that term causal emergence mean?
It's quite a simple idea, actually, and quite a sort of familiar idea. And for this reason, I find it a bit puzzling that there are some researchers who have a real aversion to it, because all it really means is that larger scale structures emerge that you can't anticipate by looking at the component parts. And the classic example of this is the murmurations of birds, or the swarming of other animals, but murmurations in particular, because we can see, when you see one of these amazing sort of flocks of birds, there's the coherence of the whole flock is astonishing as they go through these very sort of complex convolutions, and you would not be able to anticipate that sort of behavior or that sort of emergent structure by looking in incredible detail at the anatomy and the physiology of a starling. It's something that comes out from the interactions between the components. So that's really all it refers to: the emergence of some kind of structure that can't be understood from the from the individual components. The causal element of it is also to say that causation itself can be like that. So, the causation it doesn't necessarily start with the individual components. That there's causative power in the system at higher levels than the bottom ones. And I mean, this is but I suppose that maybe I should say a bit more about that, really, because what I mean is, if we're trying to look at the whole and understand how did that happen, how did this murmuration appear, how did this living organism appear, we want to think in terms of causes. What caused this to happen? And causal emergence says that if we can quantify, which we can do, there are ways of quantifying causation. There's, it's still kind of much debated, and there are many ways of doing it. But if we try to quantify it, we find that there is more causal power, more causation, literally, at a higher level of the system than at a lower one. So that's causal emergence.
Yes. I learned a really interesting example of this recently on a podcast with Dr. Earl Miller from MIT. He taught me about mixed selectivity neurons. So I always knew that, or I guess I had an idea in my head that like a neuron held some concept in some abstract way. So there was a neuron that, I'll use high level concepts to understand, but this isn't how it works. But so you'd have a neuron for car, and then you'd have a neuron nearby that holds the concept of something like a tractor. And then you'd have another neuron that might hold the concept for something like a farm or something. And then those would all be connected because like a tractor is sort of made out of the farm and the car neurons. So this is actually not how it works, but this is how I used to think about it. And he said that neurons can hold completely different concepts that are entirely unrelated and mean nothing to do with each other. So the same neuron. And then he went on further to say that actually it doesn't really make any sense to even think about a concept being encoded within one neuron. It only makes sense within the network. So if a neural network seems to be encapsulating some idea like a car, you can't break down those neurons and say this neuron's a wheel and this neuron is a windshield and this neuron is a wind wiper, whatever it is. That's just not how it works. And you have these activation patterns of all these different assemblies which encode entirely different concepts, which I didn't know. I thought concepts were very grouped together in very structured ways so that concepts are kind of, anatomically mapped in a way that made sense. But you get these assemblies that look very similar to each other, but encode entirely different concepts. And again, the idea is that the amount of variables here are related to timing, related to context, related to, there's just so many different more variables that the system can play with here that you might not be seeing in your sort of low resolution EEG readout or anything else. So no matter how much you stare at one neuron, you're not going to figure out what the neural network does, basically, is I think a decent an example of causal emergence.
Yeah. And, exactly that same process is true of gene networks and of protein networks, of the interactions that they have between them. So, there are some proteins in the network of interacting proteins that again seem to essentially encode different functions in the same protein. It can, so there are some proteins that do quite specific jobs, but they might be, one or two. There might be two or three quite specific jobs that seem entirely unrelated to one another. So
Yes.
the proteins identified for one thing, and then it turns out to do, to catalyze a completely different reaction. And these are often called moonlighting proteins, which is a kind of revealing term in itself, because we're almost saying, hang on, you're not meant to be doing that. You're meant to be doing this job, whereas, because that's the kind of model.
That's funny. I didn't know that term, moonlighting.
And whereas in fact, the living organism doesn't care at all. It doesn't sort of think, oh, I'll use this protein to do this. So but there are other proteins, and they're often the proteins that are the most highly connected in this network of molecular interactions in the cell, that you can't even talk about them as moonlighting. They do all sorts of things. They interact with maybe dozens or even hundreds of different components in different contexts. So it makes no sense to talk about them as having a role. In fact, these are often key proteins in disease. P53, the transcription factor that is, implicated in, or in some way or another, in many, many different cancers, turns out to be, we talk about it as a tumor suppressor, that it's there to suppress cancerous tumors. I don't know, it feels a little bit like that's like talking about a car as a kind of fly killer or something, because it's got I mean, it's a bit more sort of intentional than that, I think, but it's talking about one thing that it happens to do, but actually it turns out to be involved in many different processes in the cell, not just the cell cycle, which is the thing that if it breaks down, it can lead to cancer. And this is seemingly by design. And this is where, I think it becomes even harder to think about than the case of brain networks, where, we have this notion that no neuron is kind of born, if you like, created to do a particular job. It sort of finds it, in the course of our lives, of our cognition. But we think of proteins as, well, evolution has designed those to have a very particular sequence and structure and function, but it hasn't. And in fact, P53 is one of those that has a rather open structure. It's what protein chemists call disordered to a degree. It's got some bits that are sort of folded and closely packed, and other bits that are more loose and floppy. And it's that floppiness that allows it to, have this versatility in the jobs that it does. This is clearly a design feature. It's not a bug, because we see more of this disorder and more of this protein floppiness in complex organisms like us than we do in simpler organisms like, yeast, or certainly like bacteria. So it seems that evolution has, so to speak, found it a useful design principle to give proteins this sort of openness of structure and function so that they can find new roles. And, whether that's new roles sort of in the moment or actually new roles in evolutionary terms, and both seem to happen.
Yeah, like the more I learn about biology, the more I realize that brain-like activity is happening everywhere. And like we used to think about the brain as doing cognition, all these neurons wiring up with the environment, and it really does seem like that's happening at every single level of biology, like even molecular networks and genetic transcription regulation networks. Like all of these networks almost seem to be wiring up differently in responses to our environment in a very brain-like way that maybe we didn't really conceptualize it before. Like, is it possible that that's why there's so much variance here, and that my gene regulatory network is actually wired like in a foot? And wired, again, is a metaphor that doesn't work properly, but like if you were to compare and could fully compare a network in my body and a network in your body, that they might just function differently because the way they were sculpted to our environment and our development.
Yeah, I suspect you would actually find different wirings, again, in inverted commas, in different cells of your own body. I think there will be diversity, even at that level, because and that's precisely why I think the cognitive way of thinking about how life works makes more sense. That, whether or not we want to see that just as a metaphor, or whether we want to see it as more than that, whether there is a genuine kind of cognition, even if it's not cognition accompanied by awareness, I'm not too worried about where we sort of draw that line. But I think thinking in cognitive terms about how these molecular systems works makes a lot more sense than thinking about them in terms of some kind of hardwired circuit diagram.
Yes.
And again, it seems, that this makes sense when you think about what it is that organisms have to do, that they have to have this ability to adapt to their circumstances, to adapt to their context, and sometimes to innovate, not just at the behavioral level, but to innovate at the genetic and at the sort of molecular network level. So, that you need to have this versatility of components and this ability to dynamically rewire in order to have that flexibility as an organism.
Exactly. I shudder at the thought of what a biology textbook is going to look like in the year 2080. If you have to list out every single possible function of every gene and protein, like it's just going to be like 100 pages and you haven't gotten past one gene, because it just seems like everything can pretty much do everything. Like, not quite, but like.
Yeah, well, I think the great thing, this I hope anyway, the great thing is they won't be trying to do that.
Yeah,
In
because you
28,
can't.
they will have realized that that is the wrong way to do it, to list the things that, different genes can do, that we'll be thinking much more in terms of of system level behavior at these different levels of the biological hierarchy. And I'm convinced, I mean, I say in the book, and I don't think I would have written it otherwise, I'm convinced that there already are actually some general conceptual principles that we can use to think about how these systems work. And, the cognitive model, if you like, is one of them. And, the idea, as I've talked about already, that there is a fuzziness of molecular interactions that is, there intentionally, and that there is this tendency to work combinatorially. So molecules don't necessarily have specific functions, but they find a functionality in a collective way. That too will be one of the principles. And so once we've got away from that old idea of thinking that, genes specify molecules which specify particular functions, in this kind of circuit-like way, once we've got away from that and have new ways of conceptualizing, I think I'm a little bit cautiously optimistic that it'll kind of start to look simpler again, because we'll be, stopping trying to map everything out at this very sort of reductive level.
Yeah.
And I actually find this more and more myself when I look through the biological literature, since writing the book, that I'll see examples of work that I just kind of think that makes perfect sense. That there are these structures, sort of blobs, really, that some molecules form, often these sort of slightly disordered proteins, and often in conjunction with non-coding RNAs. They form blobs in our cells that we have ignored for not just decades, but actually centuries. We saw them long, long ago, didn't know what to make of them. And biologists have talked about them as speckles or bodies, or these vague terms, sort of stuff that, shouldn't be there and looks kind of messy. And we now we now call them condensates. And they're like liquid-like blobs of molecules that are almost like blobs of jelly, rather than the kind of precise molecular aggregates like the ribosome that we thought of. And they do all sorts of functions in cells. And one of those seems to be, they seem to be able to integrate information, because they can incorporate different molecules into these kind of gel-like networks and perform a kind of almost like a kind of brain-like assimilation of information that prompts some cell response. So, this is an example now that we recognize that these systems, are important components of our cells. They work by fuzzy rules, but we can make sense of why there should be these structures and how they do their job in a way that wouldn't have made sense in the old paradigm of precise molecular programming.
Yeah, really well said. I love all of this, and I just find it so interesting. One thing that comes to mind is it's going to get harder and harder to teach biology to kids without just, outright not telling them the truth. Because I think I agree with all of the principles you laid out there, but that's abstract and weird territory for the brain of a 9-year-old. Like, wow, how do you teach them to kind of think about biology in this abstract, fuzzy way? Well, actually, this thing doesn't really do this. It does all of these different things, and it depends on the context and depends on the time. It depends, like, there's a certain extent where that's just stuff is just too weird, right? Like, it happened with me with secondary school with chemistry, and you go into college and you realize, oh, all the stuff that I was just taught is actually wrong, and there was just kind of abstractions laid out to understand it at that level. Is that what we're going to have to do? Because I don't know how you teach all of that stuff which is all weird and wonderful and amazing, to like a 7-year-old.
Yeah.
Like, what do you even teach them?
Well, what I will say is I think that we're going to have to start thinking seriously about that. In fact, this summer I went to the meeting of the International Society for the History, Philosophy, and Social Studies of Biology. So it's kind of philosophy of biology, largely. And, they're the sort of folks who would think about this kind of stuff. And there was a session there on education and the new biology, which is all this kind of stuff I've been talking about. And it was asking exactly that question, can we teach it? If so, how?
Yeah.
So people are starting to, to see that as a question that we've got to grapple with. I do think that we need to get away from, I recognize, of course, in all teaching, you've got to start simple. You've got to find some simplifications, but I think they've got to be simplifications that aren't misleading and that aren't sowing ideas that are going to be difficult to undo, but necessary to undo at a later stage. And that's what I worry about that we've got at the moment. So, my youngest daughter came back from school not so long ago and said, we've been learning about genetics. And she drew this Punnett square. So the Punnett square is the old, people might have come across it, the old sort of recessive and dominant alleles. And you figure out, which offspring are how many are going to get this and how many are going to get that. And it's the classic way of thinking about a monogenic trait, that's governed by a gene that has a dominant and recessive form. And that was great. And, I dipped she knows me well enough to say,
I'm sure.
Shut up. Don't go into this, because she was talking about it in terms of why she's left-handed because I'm left-handed and the chances of that. And, I just happened to have read this review article on handedness and genetics. And my God, it is a nightmare. It is so complicated. So, so I didn't say anything, but I do worry. And there are, people in education who worry that this idea of teaching about genes actually sows at a very deep early level the wrong idea, the deterministic idea. And it's a sort of one gene, one trait idea for, eye color and so on. And even if you say to kids, it isn't always true. Sometimes it's more complex than this. Actually, it's almost never true. They are the exceptions, the Mendelian sort of P's and so on. Those are the exceptions. And what's interesting is that Mendel chose his traits precisely because they had those characteristics. He chose them, in a sense, to be exceptions because he could make quantitative sense of them. But we kind of forget that. So I do think that we do need to rethink it, because ultimately it worries me that these ideas they can feed through to a general sense that I think exists in society now of a kind of genetic determinism. And we're seeing this, as genomics becomes more prevalent. And when we see the emergence of things like these companies that, offer to predict the traits, the intelligence of your IVF baby from the genetics of embryos, it's really sort of building into this picture that is, if not, it's quasi-eugenic and sometimes out and out eugenic, and it rests on this simplistic idea of genes. So I think we need to think about how to do a better job of teaching this complexity. And I kind of think it may be possible if we are able to just get rid of, we really have to clear out the old ways of thinking. If we're still lumbered with the genetic blueprint, then saying, genes are really complex isn't going to help. But if we start from the organism, then I think, rather than from the genome, then I think maybe, and from the agency of the organism, then I think maybe we'll have a better way.
Yeah, no, I agree. I find all this super interesting. I constantly think about the education side and what's the most effective way to teach science. I just find that a really interesting problem. So if there is no privileged level in the biological hierarchy to understand the whole organism, is there a level that we can say has the most causal power? Is there? I think we've pretty well established it ain't genes, but is there a level that we could say this has the most causal power in the organism?
I actually don't think there is. because it's just the way it's just what we see. Sometimes we can genuinely say a gene has a causal influence on a trait. So in monogenic diseases like cystic fibrosis, like sickle cell disease, it is meaningful to say there is a genetic root, a genetic cause to that condition. I mean, even there, it can be complex in that, different variants for the cystic fibrosis gene or the, sickle cell disorder, even different variants have different physiological consequences, different amounts of penetrance is the technical term. So, we can't necessarily be sure what effects a given gene like that will have in a particular sort of genomic context. But nevertheless, it's meaningful to say there's a genetic cause to that condition. So sometimes it is. Very often, actually, I think the cause is some kind of malfunction of the immune system. I think that's a real hub of causation in our body, and particularly in disease. So many diseases, that even ones that you wouldn't think would have anything to do with immunity, seem to have some kind of root there. So, I imagine that there are going to be these hubs of causation, these particularly critical things, components of the body. But I think they're going to apply, all the way up. I mean, you can imagine for a lot of conditions, you could see the brain as a whole as a kind of causal hub. It's clearly a causal hub of behavior, and to some extent you can break that down and see particular sort of circuits, but there's only so far you can go with that as well. So no, I don't think there is. I think it's actually really important that we recognize that there is no privileged causal layer in biology.
Yes, very interesting. There's another really good quote in the book. We can move on to agency, because I think we've covered it and mentioned it a good amount, but I think it's the final one in our list to move through all of the basic pillars that we had laid out. One of the best ways to characterize living entities is not through any of the features or properties usually considered to define it, such as replication, metabolism, or evolution. Rather, living entities are generators of meaning. Explain what generators of meaning means for a biological system.
Mm-hmm. Okay, the way I look at it is this, that any organism is inundated with information. There's all sorts sort of coming to us, and we have to decide as organisms what to attend to and what not to. And some of that decision is made, in effect, by evolution. So our sense of smell is not particularly good. It's not particularly salient because smell for us hasn't been a particularly important thing for us to have to attend to. So that's already sort of done. But in our daily lives, we have to sort of attend to some things and figure out what's important. Is that noise something I should worry about, or is it something I can switch off to? So that's the challenge that organisms face. And we have, the systems for assimilating, for taking in and for integrating that information so as to come to some decision. And whether I'm talking about the whole organism, whether I'm talking about individual cells, or even at the sort of tissue level, those decisions are ones that I think can meaningfully be thought about as decisions about meaning. What meaning, what value does this information have to me? So it's about, those decisions can change in different situations. That the value of attending to the smell of food for us is more important when we're really hungry than it isn't. So when we're not. So I think that's really what living things are doing all the time, that they're integrating this information and in effect making value judgments about how to weigh those different inputs that they're having. And I do think there's a genuine sense in which we can think of the kind of colloquial idea we have about meaning, that we have, that, whether a work of art or, a piece of music sort of has meaning for us or not. I think that's only really the sort of extrapolated example of something that happens all the way down or I shouldn't really say down, but through the biological, system, right down to single cells. So we have this sophisticated neural machinery for creating a sense of meaning and for a sense of salience and for a sort of valence, whether it's good or bad. But I think that we can see something like that all the way down to single cells, even though, again, I don't believe myself that there's any sentience, there's any flicker of awareness there. Some biologists do, but I don't know quite what that would mean. But I still think that individual cells are having to say, they might automatically tend to respond to a concentration gradient of nutrient. But if there's very low moisture or if there's, heat in that direction or something, they might not do. They're having to make a judgment about sort of, the value to them of that particular signal. So that's what I mean in a biological context.
Yes. What do you think is, because would you say that a cell is even in itself a higher level meaning system? I'm curious what you think the lowest level meaning system in biology is. What is the absolute minimal ingredient list within a system required to make it an agent that is making meaning from its environment?
I think that's a great question, and I wouldn't want to be too definitive on it because I think it's it is a funny one. I mean, I mentioned these blobs called condensates, which I think are they're kind of integrators of information. So they too are probably in a sense they're having to weigh up information. I mean, the example I was thinking of was where two different types of condensate seem to be making judgments for yeast cells about which cell type to become, whether to proliferate, whether to go into a sort of mating competent state, or whether to just go into a quiescent state. And that decision seems to be made in part on the basis of how old the cells are. So, the meaning of different signals that they might get, pheromone signals or whatever, will be, their age will be factored into that.
Yeah.
So even at that sort of subcellular level, there's something of that sort going on. And I think even at the level of individual proteins, particularly if they're these, very sort of floppy proteins that could do various different things, they have various different states they might occupy, or things they might bind to. Even there, we might be able to, make a case for there being some kind of, certainly contextual judgment that they're making about which partner to bind to. So I think it's going to be fuzzy. I mean, I guess I kind of feel it feels to me more clear cut at the level of the whole cell, because that's an organism. That's something that can, replicate itself. That is something that I think there's pretty general agreement is a living thing. Whereas below that, even at the level of viruses, there's debate about, whether we can think of those as living at all. And I don't think many biologists would want to think of proteins as being living. So, to the extent that I think living things, that life itself is a kind of a meaning imbued state of matter, then I think it makes sense to think of it at the level of cells and above. Below that, it becomes, hazy, but I suspect it's still a useful concept.
It's funnily enough, that seems a little arbitrary for me. I've gotten more radical on this. The more episodes I do, I've gotten more radical on this, in that if your definition is just that it's not just, it's still complex, but that the system is taking information from its environment, noisy information, ambiguous information, and it's kind of self-generating its state based on that signal, based on something internal, and then it's changing its performance, physiological state, action, goal, behavior based on that. It feels hard to me to place that line at the level of the cell, because based on everything you've kind of said today, and what I got from how life works is I would absolutely ascribe that to a protein, and maybe even maybe even further. I had a conversation with a panpsychist 2 weeks ago, and maybe I'm a panpsychist now. I can't figure it out. But drawing the line sort of in any place almost seems like more logically inconsistent than just keep on going down, because there's some level of information integration happening. There's some state change based on that information. It's almost more that we're just kind of resistant to give those labels to those things, because we're like, no, I can't, no, I don't think a protein has agency. But the more and more I do the podcast, I'm definitely getting more radical on this, as I find it harder to know where the line is. Like Michael Levin said, if you're going to draw the line, you have to tell me why. And there's never a great reasoning for it. Only just a kind of like an innate, like, no, that's silly. But then if you kind of go past it logically, like it's a difficult one to do.
Yeah, I have a lot of sympathy with that position. I absolutely wouldn't want to make that line a hard and fast, dogmatic line. So I'm very open to that way of thinking. I mean, I suppose, like I say, I think the cell seems to be the minimal living unit, in the sense that it's something that is able to reproduce itself, which a single protein by itself can't do. So there does seem to be a kind of qualitative distinction there, although again, as I say, viruses are ambiguous in that respect. I mean, that's why I kind of think I'm not particularly interested in the question of whether viruses are alive or not. It's more important to me to think about whether they have agency or not. And it seems to me that when they are in a host, they have agency. When they are outside of that, they're pretty much inert chemicals. And that seems to be a more, meaningful distinction to make. And I think the same is true with, subcellular systems. But to me, I think possibly the more important thing is to recognize that there is a continuity of, for want of a better term, of ways of being. That if you're going to have the cell as an agential system, it's not suddenly going to spring out of something that at the subcellular level works in a completely different sort of machine-like way that suddenly creates this thing that isn't machine-like. Actually, I think that the more important thing to me seems to be to recognize that at the lower level, certainly down to individual proteins, there are going to be attributes they have that have something in common with the cell as an agential system, rather than being sort of perfectly sort of rigid machine-like molecules that somehow magically combine to give, to suddenly give this agential problem. So I think this agential entity. So I think that thinking in terms of these sort of fuzzier, contextual, responsive properties, thinking about the subcellular systems down to individual proteins, that's the key thing, that you've already got sort of elements of the agent as a cellular whole. Whether or not you've got genuine agency, you've got elements of it at that lower level.
What experiment do you think would be done where you'd say, oh, that's genuine agency in a protein? Like what is that definition for you? Does the protein have multiple potential things that it could do, and then it's changing its state based on some signal in the environment, and then you can prove that it's changing its decision, changing its behavior, it's changing its state in some way? Like, I'm curious what genuine agency looks like, and what little, because it's a distribution, right? Where no one's saying a protein is the same amount of agency as a human being. But are they categorically different things, or is it all a continuum and there is levels of agency there?
Well, the definition I use for agency, and it's not meant to be a dogmatic one, it's a fairly loose one, but it's I'd say that it's the ability to manipulate oneself and one's environment in order to try to attain some self-determined goals. And I think that cells can be seen to fulfill that. So the question would be, is there some way in which all of that could be true of a protein? How does a protein, what goals would a protein have? It's not clear to me that a protein would be able to have autonomous goals, and that it could, I mean, I could see that it could that it can manipulate its environment. I guess that's what proteins do. They're enzymes, they're catalysts. So to some extent, it has that. I could see actually that it could, in a sense, it could manipulate itself if it has this kind of floppiness. Although more often, what seems to happen is that other proteins will do some of that manipulation by, for example, sticking phosphoryl groups, charged groups on it that might, change its kind of conformation. But to some degree, it'll have a conformational landscape that it can explore. So I could see.
It's a slippery slope.
It's be interesting to explore if that definition is one that we think is reasonable for agency.
Yeah.
Could we imagine a situation in which a protein could show that sort of behavior? It's not obvious to me that it could do all of that, but I wouldn't rule it out.
Yeah, it's definitely not as crazy an idea as it once seemed, right? The more we learn about these biological systems and what they're doing, it's harder to remain very logically consistent with what you're saying and then say, but no, it can't be at the level of the protein. Because, I would argue that probably those tick boxes in some degree are all met at maybe even simpler levels than proteins. I don't know, smaller proteins. It just seems very hard to know where the line is, and very hard to also know what the experiments we need to do to figure this out. Like, what do the experimental paradigms look like where we would actually decipher this? That's also pretty hard to know.
Yeah, and I'm not at all sure that anyone would be able to come up with an experiment that everyone would agree is a kind of benchmark for agency or not, that comes up with a binary answer. I'm not sure if that would actually be a useful thing to do either. In a sense, it doesn't much matter to me if I'd certainly want to argue for agency at the cell and above. It doesn't matter much to me whether we can make a case for lower levels. I don't sort of instinctively reject that idea. It's a question of whether it's still a productive way to think about it. I mean, I think
Yes.
about that the same way as, there are some people who argue that there is a kind of awareness, a kind of, wouldn't say consciousness, but a sentience in individual cells. I'm not, I don't want to sort of reject that as, just being crazy. It's more a question for me of, does that remain a productive, a useful way to think about that sort of system?
Yes.
And I guess it's not clear to me when you have, you would have such minimal sentience. I'm not sure that that there would be much difference between sort of regarding it as, minimally sentient and not sentient at all. I'm not sure why it would be worth drawing a line there. So it's really a question of whether it's a productive way to think.
Yeah. I like again Michael Levin's sort of spectrum of persuadability, right? What can you persuade a system to do with the right tools? And I guess maybe if you were to, it's less important about the label that we give it, but it's more important on, well, what could we persuade a protein to do? Is there applications in regenerative medicine where if we fully knew how to get the protein to behave in a particular way, maybe the space of possible behaviors for a protein is actually a lot larger, particular proteins a lot larger than we think. And then we might be able to get it to do productive things. So with having the label of agent or not is less relevant than, well, maybe it can be persuaded to do useful things biologically that we didn't even think of. I don't know, but I would say that's certainly plausible.
Yeah, I think, I mean, Mike will have thought about this far more deeply than me, and I'd, certainly want to listen closely to what he has to say. That makes me think that a concept I think that is very useful in thinking about these sorts of questions is that of affordance.
Yeah.
What are the possibilities that a protein has? And I think actually it seems to me that biology is in the business of generating molecules with affordances rather than with very closely defined functions. Many of the most important molecules we have are ones that have particular affordances in the sense that they could find useful functionality. And I think, sometimes that's probably what's happening with a lot of transcription of bits of the genome that aren't traditionally thought of as genes with functions. So making all these RNA molecules, some of them won't have any functionality at all, and some of them could be usefully regarded as just noise, as just the transcription apparatus not being very selective about what it transcribes. But I think that there's a point at which we can also think it's making things with affordances. Those RNA molecules might be able to find some useful job to do in the cell, and we find examples where they really do.
Yeah. Do you shy away from using the word consciousness for any specific reason? That you just don't want to get into the debates about what it means, or you just you don't want to dip your toes in those waters? Do you not use sort of that word on purpose?
I guess, I guess I do not use it on purpose because I think it's likely to provoke a response. It's
Yeah.
not, it's kind of unnecessarily provocative, I suppose, because I think there are other ways we can talk about it, and because it's another of these terms that we don't have a good definition of, a clear definition of, and we certainly don't have a good scientific understanding of. So. I think it causes more trouble than it's worth to invoke that. Sentience is perhaps a less, controversial term to use, but even that, for to talk about the sentience of a protein, I'm not quite sure whether that would be at all helpful.
Yeah. So are you describing sentience here as some awareness of its own self?
Yes, yes, that's right. As some kind of, I suppose, really some kind of valence attached to the signals that are being received, whether it's whether is this a good thing or is this a bad thing. So it's I suppose it relates to this question of value and of meaning. Um, but exactly how it does, I'm wary about talking about, because I don't think we certainly don't have a good definition of sentience. I suppose it's more a sort of pragmatic question of, is this going to repel some people who needn't be repelled if we don't, invoke this term, and is there anything lost in, treading around it rather than claiming that things are sentient? But at the level of a cell, I freely admit I'm open-minded and also somewhat confused about how we think about the terms like that.
I think open-minded and confused could be the tagline for my podcast, to be honest. That is remarkably exactly what it's been like, and radicalized episode by episode, as I'm just like, I mean, maybe. Like, I have no idea. It's just so interesting. One area we discussed in our last podcast was these brain organoids, right? Where these question of defining sentience and awareness kind of does become important. Like figuring out what experience would even mean like for a cell, and if it's possible of having a negative valenced experience, what that should mean in terms of how we harvest these brain organoids and use them, right? I mean, so there are sort of interesting and weird areas in synthetic biology where this stuff is maybe becoming important, and trying to figure out, like, is there a negative valence there? Like, what are the important characteristics in guiding our synthetic biology as we do more and more kind of weird gene editing, weird regenerative medicine stuff? Maybe definition. I don't know if definitions become more important, but definitely like clarity of what experience is for these smaller systems, and if they are capable of negative experience.
Yeah, well, I did entirely agree that it's a question that we're going to have to grapple with, certainly in areas like that of, developmental biology and stem cell biology, and what's now being called generative biology, where we're growing these quite complex structures that look like Well, in particular, when they look something like little brains, that we have to, begin to ask these. Well, we have to be already asking those questions. So I don't think I'm ever going to be concerned about the well-being of my individual cells, or even the notion of, emotions or pain in an individual neuron. But clearly, at some point in the complexity between an individual neuron and a neural network this size, something develops, something emerges, and we don't know when and we don't know how. I've had a brain organoid grown myself from my own skin cells, and several of them actually, and I didn't feel a kind of duty of welfare towards them because I knew they were, too simple to feel that that was something I needed to worry about. But they were active. These neurons are active. They're signaling to each other. It's the stuff of thought, even if it's not thought itself. So I think it's entirely proper that that discussion is happening at that level of biological research. And the same is true for these embryo models that are being grown from stem cells that are, assemblies basically of stem cells, and that are, might be more or less specialized and that can self-assemble without ever having seen an egg or sperm cell, can self-assemble into embryo-like structures and start to develop. And that there too, we don't have a good framework for how to think about that in relation to human life and, human-like structures.
Yeah.
And I think probably the important thing to do is to acknowledge that gap, that we don't have ready-made frameworks for how to think about brain organoids or about embryo models ethically and morally, and to some extent even biologically. So, simply recognizing that it acknowledges that it's a complex question that we're going to have to find answers for de novo, really.
Yeah. What do you think are some of those other really big, weird biological areas that we are sort of, maybe without even knowing it, heading towards, but we just have no idea what the hell it's going to do? Like there's lots of things in gene editing. I spoke to somebody just this morning from Colossal Biosciences, the head of the team that is trying to de-extinct the Tasmanian tiger and bring it back to Tasmania. And he's a remarkably good speaker, clearly believes in conservation and is clearly pure-hearted in his intentions. But I'm sort of also worried about where stuff in that area could go if the financial incentives aren't, in the right place. And like, is there any of those sort of just weird stuff that's coming up in biology that makes you uncertain, that we need to start talking about now, that there isn't enough conversation on?
I think that's certainly one of them. I mean, I've got a lot of skepticism about what Colossal are doing, or about the claims they're making. Actually, that it's whether this is really de-extinction when you're
Yes.
making a genetically modified, whatever it is. But I think we're starting to, it's in the area that I've just mentioned of things like embryo models and other structures that we might grow from stem cells. I went to a meeting, Royal Society discussion meeting a couple of months ago on generative biology, which was talking about these issues. What can we make from biological matter as we get to better understand the principles that govern development, and how we can tweak that, and how we can sort of switch cells between different states. Entirely new possibilities come up. Possibilities like, are there cell states? And I'm sure actually there are cell states that could exist and could do things, but evolution has, so to speak, not discovered them or not had any use for them. There are
Right.
so many, this landscape of possible states of gene activity is so vast, it's utterly inconceivable to me that the only ones that are stable are the ones that biology is already using. So what would those be like? Entirely new types of tissue, maybe entirely new types of organ, let alone new types of organism. What kinds of morphologies are possible? I suspect that human cells, I mean, Mike Levin has looked at this as well. Human cells are probably capable of creating quite different morphologies from the one we have if we understand the developmental process well enough to be able to intervene in it. If we, can grow these organoid-like, these organoids, these organ-like systems, and can connect them together, which we're starting to be able to do by, vascular systems or, some kind of neural signaling systems, can we make a kind of schematic organism, even a schematic human, by joining together different petri dishes of, these things, they
Proper
sound
Frankenstein stuff.
fantastical. Well, or, even weirder than that, really. And they sound fantastical, but the more we understand, the more we can see there's no obvious reason why things like this should not be possible. There are, the question is whether there should be any good reason to do this. We shouldn't just be doing it out of idle curiosity. But there are, the reason these systems are being made, the reason organoids are being made, is because there are clear medical, biomedical, clinical possibilities that we might be able to address this way. But that doesn't mean that we should be able that anything goes with these embryo models. If we can start to make ones that look like quite advanced stages of embryo growth, we might be able to learn a lot about human development, about embryology, that we can't currently study because it's not ethical to do. But the more we're able to do it, then the better we're able to do that, the more difficult the ethical questions become.
Yeah.
So I think the whole notion of generative biology it opens up this idea that actually we really have no idea what kinds of living structures it might be possible to make when we don't rely on evolution itself to do this, this sort of blind search. And again, there's no off-the-shelf ethical or moral or philosophical framework for how we think about systems like that. So, there are definitely some very, very bizarre possibilities that we could imagine. There are also some important questions that one might imagine we could explore by exploring some of them. And, that combination means that there are going to be ethical, big ethical questions that we're going to have to discuss in the future.
Yeah. Yeah, it's such a strange one. Speaking of Michael Levin and his Xenobots, I'm supposed to be speaking to Josh Bongard next week, who built these Xenobots with Michael Levin, and these are like AI simulations running different combinations of Xenopus frog cells, and then extremely methodically, kind of surgically placed together, and then you get all this weird emergent behavior that these are just skin and heart cells, and then they start doing something that they called, kind of controversially, like kinematic replication, where it's just like a Pac-Man shaped blob of frog cells that will start going around a petri dish and accumulating cells that are dispersed around it, create copies of itself, which at a certain point will start also moving and create more copies. And it's like, well, that's bizarre. That's really weird. That's as you say, sort of biological systems being placed in a new space of possibilities that evolution hasn't put them in. And as we've discussed throughout here, the core here is that biology creates agential matter. And who knows what that agential matter would do in all of the possible spaces that aren't biology. And as you say as well, that there is clearly so many positives that could come from this in terms of regenerative medicine. Like, could we have full limb regrowth for amputees? I don't think anyone could say that that's not possible. I'd say there's pretty good reason to believe that the human body still possesses the ability to regrow a limb with the right sets of stimulus and the right protocol. And that's an insane possibility, but that would be miraculous for a lot of people, right? That would be life-changing. It would be an incredible thing to be able to do. But where will we bring the science on the mission of trying to do that good thing? What will we discover that is just utterly too strange and weird to even know what to do with, know what to police? And Like I don't know. Like have you thought about like how you think we should balance the regulation with the funding, and just like in any of these areas that clearly can do so much good, but also clearly have potential for misuse and could be weaponized in some ways. Like, what's even some guidelines for thinking roughly about this?
Well, I think the question of, why are we doing this? What is the potential value of doing this is a central one. And it's one to which there has to be a good answer, more than, well, I'm kind of curious to see what will happen. I mean, I think there's plenty of room for curiosity-driven research in a lot of basic biology, but there comes a point where that's not enough. If you're creating something that raises ethical questions, that there are all kinds of experiments you could imagine doing in human embryology that would be curiosity driven, but that would be ethically questionable. So asking that question, why do this? What's the potential benefit? I think is a really important sort of general criterion to be guided by.
I think you'd even get pushback on that though, right? Because it's, scientists would, I think, make the argument that, well, there's an interesting area here that's unexplored and we don't fully know where it could go, but it could lead to something very important. And that can be maybe hard to regulate at the grant process, because it could be true that the answer to some really big scientific questions could be in directions that seem kind of strange but don't have a clear hypothesis behind them. So it's weird. I agree with you, but I think some scientists, I think, would have that would be hard to agree on as well.
Yeah, I think that's true. I mean, I think it's, you're weighing up different things. So if you want to, do it and you're not quite sure what will happen and you think there might be interesting possibilities, that's fine. If you're not wanting to do it with, 28-day human embryos, for example.
Right.
But it's fine if you wanted to do it with, totally sort of experimental stem cell systems in culture or something. So yeah, I think that there's always going to sort of be that balance, and that, it shouldn't be, it absolutely is important that research isn't sort of driven purely by, well, what is this going to be used for? What are the applications? What are the benefits? And if you can't state them, you don't get funded.
Yes.
So it's, there has to be a balance. And that's why I kind of think there aren't any hard and fast rules. The things I would want to bear in mind in navigating this area are, first of all, the that we come to these sorts of questions recognizing that culturally we have preconceptions about them, and that those are going to inform the way we think very often. As a culture, they're going to inform them. I mean, I've got this, I wrote years ago this book, Unnatural, to look at that very issue, that we have deep-seated cultural ideas about what is natural and what is not, and their moral judgments, as well as purely sort of categorization of what happens in nature and what doesn't. And we need to be aware ourselves of those prejudices we bring. So I think that's really important. And I think also is going to have to be an ongoing process, because the technology is changing so fast. And, one that is able to be revised. I think that's one of the great things in the UK. We have the HFEA, the Human Fertilization and Embryology Authority, which makes decisions about this kind of work and is widely seen elsewhere as a kind of model for how to do this, that it's been able to be quite permissive about research without just being laissez-faire, without just, letting the market do its thing, which tends to happen in the commercial sector in the US. So there is regulation, but it's permissive and it's thoughtful. And in fact, plenty of researchers in these areas in the UK say that's something that's actually really valuable for research. It's not, they don't want to be able to do whatever they like. They want to have clear rules about what is and isn't permissible, and rules that can be revisited and are regularly revisited. So I think it's more about, rather than having principles, it's more about looking for good models of regulation in the past, and the HFEA is one of them. But at the same time, I know they would say, because I've spoken to folks there, it's a struggle for them now to keep track with the pace of change in these areas, that to be able to do that internally, let alone then drawing up guidelines or even sort of legal restrictions on what can and can't be done. By the time that's happened, things have moved on again.
Yes.
So it is becoming a real challenge, how we're going to navigate, this landscape as things get more and more, as you say, more and more bizarre, because we understand more and more about what biology is capable of doing and how we can intervene to make it do those things.
Yeah. Yeah. And I think another scary side is that, again, since the fact that we aren't all at peace in the war and we don't just all hold hands and sing kumbaya, is that, there's a chance we end up in another biology arms race. We're in an AI arms race at the moment where we have global superpowers competing with each other for AI supremacy, whatever that is, because they think that will give them some geopolitical advantage. Like, 20 years down the line, who's to say we don't find ourselves locked in a generative medicine sort of arms race where people are just doing synthetic biology with no regulations because the fear is that we'll lose out to some superpower in some hypothetical way because they're creating animal chimera hybrids that are bulletproof and can invade countries. And I know that's getting really science fiction and weird, but regulation tends to completely go out the window when we're competing with another country for some sort of political reason, right? And we're finding ourselves in another arms race at the moment that I don't think a lot of people agree with. And I think too much resources are being put towards it and no one really knows where it's going, and it might be bad for humanity. But because both sides are kind of justifying it on the basis of, well, we can't let China win, China can't let the US win, that if that happens in biology, that could just have really dramatically terrible outcomes as well. That's very speculative, of course, but we human beings haven't been very good at reining in that sort of behavior in the past.
No, I think that's true. I mean, I think set against that is the idea that if there are clear medical benefits to an approach, then that feels a bit different to the AI system where the AI example where it's often about who has power, who has corporate power, who has governmental power. The driver of a lot of this stuff is going to be a medical driver. It is going to be about welfare. But that said, I take your point that that if there's competition and a country wants to get ahead, that it might be tempted to be laxer on regulation. I guess all I can say is that I don't think that's happened so far. That China has regulations. We might imagine, in the West, kind of stereotypically thinking, oh, it's just a wild east in China, and you can do anything because they want to get ahead. And that's not the case. There are regulations in China and they are enforced, and we've seen that happen. And that's true in most countries. I mean, I think it's more concerning in the US, where it's not so much about that, but about, just market driven, ideologically market driven. And, the regulation is very lax. But I'm not clear that that's necessarily going to, it hasn't been beneficial, in terms of getting new research done. Actually, more has been done in the UK, in that respect. And a lot of American scientists have come here for that reason, because the regulatory environment is much more straightforward, much more, permissive in some ways. In the States, you have this weird situation of the sort of laissez-faire in the commercial sector, and really quite prohibitive, or it has been, at times in the.
Really, I wasn't aware of that.
Well, things like the restrictions that the Bush administration put on embryo research, that it couldn't be funded with federal funds. It was really, really restrictive, and I've lost track of exactly where it stands now, but I think it still is more restrictive than it is in the UK, for example. So, in some ways, I think it might be hard to generalize about where it might go, because the specifics of, that country's political system and, economic system might sort of partly dictate that. But I think also, again, it's possible to sort of, start imagining these sort of science fiction scenarios for why a country might, the old one was when cloning was around in the 1980s, 1990s, it was people are going to clone Hitler. It made, I mean, really, it was literally, imagine an army of Hitlers. Well, imagine it, this guy with, who was really quite a weak, feeble person. What does that even mean? But that seemed to be a real concern that was coming up. So again, I kind of think it's, really important that we keep an eye on our kind of fantasies and our cultural obsessions with these sorts of ideas, which the new biology and these new biotechnological possibilities definitely tap into, but they're not actually replicating that.
Yes.
Yeah.
No, 100%. For the last sort of 10 minutes of a podcast, I like to dip my toes in the really bizarre stuff, just to get people's opinion, just to get the really people in the know, like what are they cautious of that could come down the line. But this has been a truly incredible conversation. Every time I speak with you, you blow my mind in how good you are as a scientific communicator. I've loved this conversation. I think it's such a good synthesis of modern biology, and I think the pillars that we went through are such good lenses for understanding what biology actually means. I think we've debunked a bunch of myths that probably a lot of the listeners would have held in their minds about how biology works. I think everyone should absolutely go read How Life Works. It is really definitely one of the best books I've read this year, probably one of the best books in general I've read on biology, with how accessible it is, and again, just how good a job it does at synthesizing really murky work that doesn't often seem to even line up, like all these different results that don't seem to come to a grand consensus or any grand conclusion. Like, it's very good at laying that out. So as always, thank you so much for coming on the conversation, and I love our conversations. I'm excited for round 3. I love these so much. Philip, thank you so
totally
much.
Up for that, Ever. No, I knew I would, from past experience, I knew it would be a great conversation to have. So I've really enjoyed it. And yeah, feel free to ask me again. Happily.
Thank you, Philip.
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel. Check out our 26 Neuroscience book. You can download it for free